Well, good evening, everyone, and thanks so much for joining me tonight for an Innovation Nation. So tonight we're going to be talking with Dr. Sean Feast, and Sean is the founder and CEO of Precision Chroma, and he's also an assistant lecturer at the University of Canterbury. And Precision Chroma is a spin-off company based at the University of Canterbury that has developed unique patented 3D printed chromatography columns. Now, chromatography is a laboratory technique used for the separation of a mixture into its components. So it's going to be really interesting to hear from Sean with he and his team incorporate a 3D component into the mix. Now, Sean is also a finalist at the High Tech Awards 2023, which is taking place this Friday in Christchurch, and I will be there to see. Now, I just will mention too, as I dropped in to see Sean recently when I was visiting the University of Canterbury, because they have a real innovation hub there. But anyway, thanks for joining us tonight, Sean.
Kia ora, Judith. Thank you very much for having me. It's an absolute pleasure.
Well, look, can you start off with a little bit of your background of your tenure at the University of Canterbury and transition into becoming a founder and CEO of Precision Chroma?
Yeah, I've actually been a long stay at the University of Canterbury. I had the pleasure of doing my undergrad in chemical and process engineering, and then I went away to industry and worked for a year. And it was part of that work that led me back to the university. I wanted to get into R&D as a part of that job, and a lot of the people they employed for R&D had a further qualification, master's or PhD. So that led me back to the University of Canterbury, and I jumped on the 3D printed chromatography project, which was in its very early stages. And I was only planning to do a master's at that point, but I fell in love with the project and quickly converted to a PhD. And then once I finished my PhD, I still wasn't finished with the technology. I can really see the commercial benefits of it. So I stayed around for a postdoc. And in that time, I spent a lot of my time working on how we were going to commercialize the technology, and also lecturing at the University of Canterbury as well.
Well, say with chromatography, not a word that naturally is spoken about all around Parliament, but can you please explain what this laboratory technique is and what is it used for?
Yeah, of course. It's quite a mouthful of a word. In its essence, it's about separating out a mixture of components into their individual molecules. So it can be really useful for scientific research, where you have a mixture of substrates that are either biologically based or chemically based. So things where you have a mixture and you want to separate them out into individual components. We use this particularly for biologics, where you have a soup of materials, proteins, DNA, cells, and you're interested in one of those proteins. So to analyze it further or to use it for things like therapeutic drugs, you need to remove away everything else from it. So chromatography is a really good technique for isolating just one component from a big mixture.
And the 3D chromatography, that's an innovative technique in which you've developed, and it's paving the way in the next generation of chromatography. So how does that work? I mean, it sounds fascinating.
Yeah, it's a really innovative technique. So a traditional chromatography approach makes use of little beads, really, really small beads, and they're packed randomly into a column. And then you can flow a liquid through the gaps in those beads to act as a filter. So if you imagine sand on the beach, you can pour water over that sand and the fluid will just flow between the random gaps between those sand granules. So that's how a traditional chromatography column works. What we're doing with 3D printing is, instead of having these random flow channels between little beads, we're actually designing the flow channels using chemical engineering principles. So in chemical engineering, you never want something that's random. That leads to a whole bunch of errors and a whole bunch of possibilities for mistakes. You want to know everything about the structure you're working on. So we can design these flow channels to be perfectly accurate, to know the exact shape of them, how many there are, and the size of them as well, which is really important for chromatography. So we're taking advantage of the ability to print exactly what we need for chromatography and use it for a chromatography column.
So this is fascinating, and I imagine there's been a lot of research and development that's gone into this unique method of chromatography. But can you tell me a little bit about that whole R&D journey? I mean, the idea of saying this is what we have been doing for years, and now we're going to 3D print it. I mean, it's just extraordinary. So how did that come about?
Yeah, so my supervisor at the University of Canterbury originally had the idea of being able to design flow channels instead of using these randomly packed beds of beads. So in chemical engineering, these random packed beds of beads are the gold standard. And he had the idea of, well, what if that wasn't the case? What if we could design these flow channels? And that was probably nearly 10 years ago now. So there's been a whole bunch of work that's gone onto it. So it originally started as an idea, and then it went through a whole bunch of computer-aided analysis using fluidics and computational analysis. And then once 3D printing, the process caught up, we were actually able to make these structures instead of just imagining them and designing them on computers. And then from there, as the 3D printing process got better and better and better, we were able to make smaller and smaller structures. So the channels we're looking at are about 1/10 the size of a human hair. So really, really small. And as 3D printing got better and better, we were able to make these structures a reality. And now the process is so good, we're actually scaling it up and getting towards a commercial scale column.
Well, that would be great, wouldn't it? In many ways. So look, how many are working with you in this, in your Precision Chroma team, because and working on this technology?
Yeah, so we're quite a small team. There is myself, Fabian Dollamore, my lead engineer, and Vivek Menon, who is my research assistant. But outside of the core team working on the technology, we have a lot of support from the university. So Professor Conan Fee is still heavily involved with the company as a scientific advisor. And we also have great support from our first investors, which were Bridgewest. So they provide a lot of the input into the business side of things, and they've got a global network in biotech, which really opens the doors for us to be able to engage with large pharma companies, large life sciences companies, and also, really provide some of the help on the business side of things.
Well, it's a perfect example, isn't it, of great research going on in New Zealand at our universities, then moving into a startup, and then bringing in partners to help you actually go global. Because as we know, there's no point being the very best in New Zealand when you've got this sort of work. You really need to be global, and what a great place to start it from, and as your launching pad. So, what sets 3D chromatography apart from traditional chromatography methods? So, what's the difference in the methods?
Yeah, so it's all about that design structure. So like I talked about previously, all those random granules have gaps between them that allow fluid flow. But those gaps can be small, they can be large, and it all depends on that random nature. So for a traditional chromatography column, you have to be really careful about what you put over that structure. It has to be free of solid particles. It has to be very, very clear to make sure nothing can get caught in those really small gaps. So what we can do with our 3D printed chromatography columns is design those channels to be large enough to passage solids. So how our technology is beneficial for the industry is we're able to do a number of processes, not just chromatography, on our chromatography column. So we can do the filtering process, the removal of cells, cell debris, and solids. At the same time, we can do the chromatography process. So we can separate out big stuff, medium-sized stuff, and small stuff with our chromatography column, where a traditional chromatography column can only passage small stuff, therefore it can only separate out the small stuff. So we're combining about 3 different processes into one, saving time, consumables, and capital equipment costs as well.
Well, this is fantastic. And you've really answered my next question, which was about the benefits of this, because basically it's a far more precise result, isn't it?
Yeah, we're essentially what we're trying to do is simplify the way biological pharmaceuticals are manufactured. So those being things like mRNA for vaccines, proteins like insulin, gene therapies as well that can really save lots in healthcare costs as well. So we want to shorten that manufacturing process by combining those three steps into one. Making it cheaper to do as well. So there's a whole bunch of follow-on effects. So all the way from when you're discovering a drug, you want to be able to discover it, check that it works, go through all those clinical trials. But each time you go through each one of those steps, you have to manufacture some of that drug. So by shortening that process down, all these steps combined shorten that entire process. So it's about getting drugs to market quicker, as well as getting the manufacturing costs down as well.
Well, that has got to be good. So is this technology commercially available at the moment to be used?
We're currently working on our first product to bring to the market within the next year. So we're currently working with some commercial partners overseas, testing the technology and helping us develop it to its first product. So there's a couple of things there where we're scaling it up, making it larger for commercial manufacture, as well as making sure that the people that are going to be using it in the end are happy with it and will be wanting to implement into their process. So we believe within the next 12 months, we'll have our first product on the shelf.
And I guess that your market is going to be the pharmaceuticals in a big way.
Yeah, so that's the first one. So the pharmaceutical market is one of the largest in the world, and that's going to be our first market. But we're also hopefully going to see use cases outside of that pharma market into things like precision fermentation, which is the manufacture of proteins such as enzymes that are used in a whole bunch of different industries, and also potentially in alternative foods as well. So pharma's the first, but there's a lot of use cases for our technology.
So if people are interested in the technology or getting involved in some way, whether it's to assist with, in some way they might be interested in, how would they get in touch with you, Sean?
Yeah, so you can reach out to me on LinkedIn or via our website as well. So precisionchroma.com. That's probably your first port of call. But if not, look me up on LinkedIn, Sean Feast or Precision Chroma, and we'd be happy to hear from anyone who's interested in using the technology, interested to hear more about what we're up to. We are currently raising capital right now as well. So looking for future investors in our technology to bring it to the global stage.
This is great. So lastly, Sean, you're up for an award this Friday at the High Tech Awards dinner in Christchurch. And what is it that you've been nominated for? In what category?
Yeah, I've been very fortunate to be a finalist in the Zero Young Achiever category. So I'm up for that on Friday and really excited to be in a room with New Zealand's finest as part of the High Tech Awards. It's a great celebration of all the amazing companies that are in New Zealand and the amazing stuff we're doing. I think people probably underestimate how much of an impact New Zealand has on the global stage in terms of technology. So it'll be a really great night.
So, Sean, since it's Young Achiever, how old are you for our audience?
I'm 28.
So 28. And all of your tertiary education has been here in New Zealand?
Yes, all at the University of Canterbury, yeah.
So all at Canterbury. You did your bachelor's, your master's, and now your PhD. You've done that as well. So, yep. I mean, wow, that's so impressive. And thank you so much, Sean, for spending some time with me tonight and with our audience, because this is really cutting edge stuff. And when we think about the opportunities to go global on this sort of research, they're mammoth. And I expect you're to do very well. And when you are very well, will you still come on my show?
Oh, definitely. Yes, without a doubt. I'd love to come back and talk some more. Hopefully when we are selling into overseas markets and we're a massive company based right here in New Zealand.
Oh, so happy for you, Sean. And I'm sure your whole family is very proud of you as well, as long as they are aware of what it is that you're doing, because I've just got a feeling you probably spend most of your time working.
Yeah, I do spend a lot of time working, but my friends and family definitely support me along the way, and I know a lot of them are probably listening right now. So
right.
it means a lot to have them there, and they all listen to me waffle on about chromatography all the time. So it's awesome.
I think it's a very interesting subject, and thank you, Sean. And I'm very pleased that I popped in to see you in your lab when I was recently in Canterbury, and really thank you for this evening. This is very, very. I say it's not only groundbreaking work, but it's global, and that's where we need to be. Thank you, Sean.
Thank you very much, Judith. Thank you very much for having me. It's been a pleasure.
Thank you.